Hello and welcome to Professor Gadgil GPAT Academy. Today we will discuss about Krebs cycle. It is also known as the citric acid cycle or the tricarboxylic acid cycle. Imagine you have just eaten a meal. The carbohydrates are broken down to glucose, fats, to fatty acids, and proteins to amino acids. All of these diverse fuels converge into a single central metabolic pathway inside your mitochondria. That pathway is the Krebs cycle. It's the final common route where the carbon skeletons of sugar, fats, and proteins are oxidized to carbon dioxide, and in the process, the cell captures high energy electrons to make the ATP, that is, the energy currency of life. The cycle was discovered in 1937 by Sir Hans Krebs, a German born British biochemist. He pieced together the cyclic nature of these reactions by studying pigeon breast muscle, a tissue with a very high metabolic rate. For this discovery, he was awarded the Nobel Prize in Physiology or Medicine in 1953. The cycle is now registered as one of the cornerstones of the cellular metabolism. Where does this happen? In eukaryotic cells, all the enzymes of the Krebs cycle reside in the mitochondrial matrix, the gel-like space inside the inner membrane of the mitochondrion. The only exception is the succinate dehydrogenase, which is embedded in the inner mitochondrial membrane itself, serving as a direct link to the electron transport chain. There are total 8 steps. The cycle begins with a 4-carbon molecule, oxaloacetate, and a 2-carbon unit, acetyl-CoA. With each turn, 2 carbons enter and 2 carbons leave, regenerating oxaloacetate. It's a closed loop, a true cycle. Let's walk through each of the 8 enzymatic steps. I'll describe the chemistry in a way that highlights the logic of the reactions. Step 1 is the condensation or the entry point. The enzyme is citrate synthase. Reaction is the acetyl group from the acetyl-CoA is transferred to oxaloacetate, forming the citrate and releasing CoA. Why it matters? This is a committed step of the cycle. Citrate synthase is an allosteric enzyme that controls the rate of the entire cycle. The reaction is highly exergonic and essentially irreversible. Chemical logic: the methyl group of acetyl-CoA is relatively acidic. Oxaloacetate's carbonyl carbon is electrophilic, and aldol condensation occurs, yielding cityl-CoA, which is then hydrolyzed to citrate. Structure inside: citrate is a symmetrical molecule with 3 carboxyl groups, hence the name tricarboxylic acid cycle. Step 2 is the isomerization, that is, rearranging the molecule. Enzyme is the aconitase, more formally aconitase hydratase. Reaction is citrate is isomerized to isocitrate via a dehydration-rehydration mechanism, passing through the intermediate cis-aconitate. Why it matters? Citrate's tertiary alcohol group is poorly positioned for oxidation. By moving the hydroxyl group to an adjacent carbon, aconitase creates a secondary alcohol perfectly set up for the next oxidation step. Next is the chemical logic. Aconitase contains an iron sulfur cluster that stabilizes the transition state. No coenzymes are needed. Step 3 is the first oxidative decarboxylation, which is the rate limiting step. Enzyme is isocitrate dehydrogenase. Reaction is isocitrate is oxidized and decarboxylated to alpha ketoglutarate. NAD plus is reduced to NADH and CO2 is released. Why it matters? This is the rate limiting step of the cycle. The enzyme is strongly activated by the ADP and the calcium and inhibited by the ATP and the NADH, making it the key regulatory point that matches the cycle activity to energy demand. Chemical logic: oxidation of the alcohol to a carbonyl forms an unstable beta-keto-acid intermediate which spontaneously decarboxylates. Mg2 plus or Mn2 plus ions are required for the activity. Step 4 is the second oxidative decarboxylation, that is, another CO2 is lost. Enzyme is alpha ketoglutarate dehydrogenase complex. Reaction is alpha ketoglutarate is converted to succinyl-CoA. Another NAD plus is reduced to NADH. Another CO2 is released and the CoA is covalently attached. Why it matters? This is a massive energy complex, very similar to the pyruvate dehydrogenase complex. It uses the same five cofactors: thymine pyrophosphate, lipoic, lipoic acid, CoA, FAD, and NAD+. Clinical note: arsenic poisoning inhibits this enzyme by binding to the lipoic acid cofactor, blocking the cycle. Step 5 is the substrate level phosphorylation, that is direct ATP or GTP synthesis. Enzyme is succinyl-CoA synthetase, also called as succinate thiokinase. Reaction is the high energy thioester bond in the succinyl-CoA is broken, releasing CoA and driving the phosphorylation of GDP to GTP. Succinate is formed. Why it matters? This is the only step in the entire cycle where ATP is directly produced, hence substrate level phosphorylation. In our cells, the GTP can be readily converted to ATP by the nucleoside diphosphokinase. Step 6 is the oxidation, that is FAD enters the scene. Enzyme is succinate dehydrogenase. Reaction is succinate is oxidized to fumarate and FAD is reduced to FADH₂. Why it matters? Succinate dehydrogenase is the only enzyme of the cell that is embedded in the inner mitochondrial membrane. It is actually complex 2 of the electron transport chain. The FADH₂ produced feeds electrons directly into the ETC, bypassing the complex 1. Chemical logic: the oxidation of a single carbon-carbon bond to a double bond is not strong enough to reduce the NAD+, so FAD, with its higher redox potential, is used instead. Step 7 is the hydration, that is water across a double bond. Enzyme is fumarase or fumarate hydratase. Reaction is water is added across the double bond of the fumarate to form the L-malate. Why it matters? This is stereospecific reaction, sets up the final oxidation. Only the L isomer is produced, which is essential for the next step. Last step is the regeneration, that is, back to oxaloacetate. Enzyme is malate dehydrogenase. Reaction is malate is oxidized to oxaloacetate, and the third and the final NAD plus of the cycle is reduced to NADH. Why it matters? Oxaloacetate is now regenerated and ready to accept another acetyl CoA, closing the loop. The equilibrium of this reaction actually favors the malate. However, the cycle pulls it forward because oxaloacetate is continuously consumed by the citrate synthase and the NADH is reoxidized by the electron transport chain. Next, we will discuss the energy tally. Now we will discuss the molecule and the quantity per turn. NADH quantity per turn is 3, FADH₂ quantity per turn is 1, GTP or ATP quantity per turn is 1, and CO2 quantity per turn is 2. Each NADH can yield about 2.5 ATP via the electron transport chain, and each FADH₂ about 1.5 ATP. So the total ATP equivalent from one turn of the cycle is roughly 10 ATP. For one molecule of glucose, the cycle alone yields the 20 ATP, and that's on the top of the ATP from the glycolysis and the pyruvate dehydrogenase step. How is the cycle regulated? The cycle is exquisitely turned to the cell's energy status. The key control points are citrate synthase, isocitrate dehydrogenase, and the alpha ketoglutarate dehydrogenase. Citrate synthase is activated by ADP and NAD+, and inhibited by ATP, NADH, citrate, and succinyl-CoA. Isocitrate dehydrogenase is activated by ADP and calcium, and inhibited by ATP and NADH. Alpha ketoglutarate dehydrogenase is activated by calcium and inhibited by NADH and succinyl-CoA. When energy is low, the cycle speeds up. When energy is abundant, it slows down. It's a beautiful feedback system. Now we will discuss the amphibolic nature, that is, more than just the energy. The Krebs cycle is not simply a catabolic pathway. It is amphibolic, meaning it serves both in the breakdown of the molecules and in providing the precursors for biosynthesis. Citrate can be exported to the cytoplasm to provide acetyl-CoA for fatty acid synthesis. Alpha ketoglutarate and oxaloacetate can be transaminated to form the glutamate and aspartate, respectively, the building blocks for amino acids. Succinyl-CoA is used in the synthesis of heme, that is the oxygen-carrying part of the hemoglobin. Oxaloacetate can be converted to glucose via the gluconeogenesis. So the cycle sits at a metabolic crossroads, constantly balancing energy production and biosynthesis. Now we will discuss the clinical and pharmacological relevance. Understanding the Krebs cycle is fundamental to grasping many disease states and drug actions. First is the beriberi. Thiamine or the vitamin B1 deficiency impairs the alpha ketoglutarate dehydrogenase complex, which requires TPP, leading to reduced ATP production and neurological symptoms. Next is the arsenic poisoning. Arsenite binds to the lipoic acid cofactor of the alpha ketoglutarate dehydrogenase, inhibiting the cycle. Next is the fluoroacetate, or rodent poison that is metabolized to fluorocitrate, a potent inhibitor of the aconitase. Next is the cancer. Mutations in the isocitrate dehydrogenase are found in certain brain tumors and leukemias. The mutant enzyme produces 2-hydroxyglutarate and oncometabolite that promotes tumor growth. Last is the fumarate deficiency, a rare genetic disorder causing the accumulation of fumarate, leading to severe neurological impairment. So, this was everything you need to know about the Krebs cycle. If you found this video interesting, do like, share, and subscribe. Download our GPAT Pro app for your ultimate GPAT, NIPER, and government pharma exam preparation. Stay tuned for more videos. Professor Gadgil signing off.